Welcome back to the Business of Biotech. I'm your host, Ben Comer, Chief Editor at Life Science Leader, and today I'm speaking with Sean Ainsworth, CEO and Chairman of the Board at Immusoft, an autologous B-cell therapy company developing redosable treatments that don't require immune suppression or myeloablation. We'll talk about Sean's journey from the Navy to the lab and into entrepreneurship, his founding of RetroSense and early development work in optogenetics, what he's learned from the acquisitions and deals he's been a part of prior to joining Immusoft, and we'll get into Sean's approach to building teams and raising capital and the opportunity to improve upon enzyme replacement therapies with engineered B cells. We'll also get Sean's thoughts on the regulatory outlook for cell and gene therapies. Thanks so much for coming on the show, Sean.
My pleasure. Great to be here, and thank you, Ben. Appreciate it.
Glad to have you. You got your undergraduate degree in microbiology at the University of California, San Diego, and were in the Navy before that. How did you get interested in microbiology initially?
Great. Thank you. Yeah, I actually, going back to my high school days, that was just always my favorite subject was biology. As you mentioned, I did a stint in the Navy right out of high school. I guess I still needed to figure out what I wanted to be when I grew up, one might say. After, through the Navy, I did get the GI Bill and put myself through college with the GI Bill. It was a natural uh, progression for me to get into biology and UCSD was just, the perfect school for that, having been in the Navy in San Diego.
Right at the base there, I assume.
Correct. Yeah.
You did some lab work on stem cells, gene modification to stem cells in San Diego. Can you describe, what did you learn from that experience?
Yeah. Thanks. It was really exciting science for me, and that's what drew me into that particular role. One might actually think of it as kind of an early attempt at gene modified cell therapies. We were looking at modifying stem cells from cord blood to effect proliferation without differentiation. And so the science was just immediately compelling to me. I learned after a few years, that it's the old expression, 1% inspiration, 99% perspiration. I spent a lot of time at the bench doing pipetting, running PCRs, counting colonies and plates, and reached a point where I realized I didn't want to do that for my whole career, and so decided to go to the business side at that point.
Well, tell me about your first experience with entrepreneurship and maybe what the outcome was.
Sure. Yeah. So that, fast forwarding a bit, I started a company co-founded out of University of Michigan that was called Compendia Bioscience. And Compendia was a gene modifying, pardon me, cancer gene expression profiling tool. Yeah, we launched that. That goes back to 2006. The company was ultimately acquired by Thermo Fisher and $50 million acquisition, but the outcome was good for everybody. We never had to raise a lot of money for that, a couple $1 million. So that was a nice outcome. Uh, and yeah, that was an exciting endeavor.
So you went to the lab to starting your first company and getting a nice exit. That's, a pretty good first experiment, on the entrepreneurial side. Where did you get the skills, that were needed to make that happen? Assembling a team, raising capital, getting to that exit point.
Yeah. Well, I actually there was a bit of work in between the lab and my first entrepreneurial endeavor. So I worked actually in an intellectual property law office in Tokyo for a couple of years. My brother was an IP attorney, and during back in those days, we used to chat a lot about our respective work, and I was definitely intrigued by the whole um, concept of intellectual property and what how that applies in science and biotechnology most specifically. So from there, I went to business school at WashU in St. Louis and got into corporate development consulting. And so that kind of brought in the intellectual property experience, the lab experience, and the international experience living in Japan. So that work was predominantly focused on the Asia Pacific market, but it did give me, a lot of experience with, the concepts and the ins and outs of licensing and intellectual property. And so that led to that first endeavor of licensing a program out of University of Michigan and that first startup.
And where did the funding come from that? I mean, were you also kind of building a network of investors and meeting, VCs and stuff like that at that time as well?
To some degree, yeah, yeah. So I'll start by saying we actually had our first sale before we were established as a company. So I had to run out and open up a bank account to deposit our check, and we had to jump through some hoops to to make all that happen. So it's always nice having revenue, at the outset. But indeed, we did. I'll give credit as well to. The local financing, Michigan Economic Development Corporation, funded us for, I want to say, $2 million, and then we also did raise $2 million in investment capital.
Okay. All
Yeah.
right. And so you had this exit with Compendium. What did you do next?
So I started a company in the meantime called RetroSense Therapeutics, and RetroSense was the really was the first optogenetic approach to enter clinical trials. So that was a gene therapy for vision restoration in retinal degenerative disorders. We took that into
Yeah,
the
and that's a
clinic. Yeah,
no,
go ahead,
yeah,
please.
sorry, I didn't mean to I cut you off, but that was a big white space at the time, very early company working in optogenetic therapy. And I mean, was that you, certainly it was a risk, but how did you decide to select that particular IP for RetroSense and, take a kind of a big swing in a brand new area?
Yeah, great question. Thanks. I mean, it's almost like it chose me. So the story there is, the IP came out of Wayne State University, and they were doing some evaluation of a number of different programs and trying to assess whether, it made sense to protect the intellectual property. And they like to talk to industry folks to get, some sense of where things have sticking power and not. And I met with the inventor, Dr. Johua Pan. He presented his data, and I was floored. I went home. I actually was chatting with my wife about it, and I said, if this is, if this actually works, this is one of the most amazing things that I'll probably ever see in my career. And so, yeah, I long story short, I got back to the Wayne State tech transfer folks. I said, yeah, this is, you definitely need to protect this intellectual property. It's going to be important. And they came back to me and said, can you help us, build out the development plan and the business case? So I actually worked on that for about a year, and I hadn't planned on jumping back into the entrepreneurial side of things, but I knew I would never find anything as interesting, exciting as that. And so, yeah, I took the plunge that second time, uh, and ultimately, I think we were acquired by Allergan after dosing our first patient. So,
Yeah, two for two on, being acquired on companies that, that you work to form and launch. The in addition to working in a really innovative space and new space at the time, the other challenge you faced was a broad kind of macroeconomic situation. This is 2008, 2009, I believe,
yeah.
where, kind of similar to some recent years that we've had in terms of the investment sentiment and situation in life sciences. But what could you say, Sean, about how you built the team at RetroSense during a really difficult economic period, and how you kind of secured the funding to get you to 2016 and the Allergan acquisition?
Yes. So the building of the team, I think the first and foremost was finding people that shared the same passion. So we were a small team, but everybody that was on board really understood the importance of the science, the potential impact to these patient populations. And so that was kind of rule number one for me is let's find people that really get the importance of what we're doing, the why, and really have share that passion with me. So indeed, we did build out the team. We did have some rough years. As you point out, 2009 was still kind of a downturn in the market. And looking back, there were no gene therapies even approved in the Western world at that time. So I had initially expected that, the first would have been approved in 2010, and that got bumped out to 2012. So we started out with some angel investors that kept the company moving forward. And again, credit to the Michigan Economic Development Corporation and some of the other local funds. They kept us able to move the science forward, do some of those important IND enabling studies, which are not hugely expensive. And then we started getting institutional investment after, in that 2012 timeframe. Once that first gene therapy was approved in the Western world, and there was a lot of excitement then around that time surrounding gene therapies. So we still didn't raise a tremendous amount of capital, about 15 million all in. So by today's standards, that's still a small round, but that was sufficient to take the company through to acquisition.
There's a common refrain in the industry. A number of guests have said it on the show. We've published a byline piece fairly recently that had it right in the headline. Biotech companies are sold, not bought. Can you talk a little bit about, how you kind of set up RetroSense to even be an acquisition target for Allergan, and how you thought about that?
Sure. Yeah. I think that any, responsible biotech entrepreneur has got the end in sight before even founding the company. And so, not to say that I knew Allergan would be the buyer, but you're building to be either acquired or taken public. And so part of my every day was talking to industry, understanding what they were interested in, what they were looking for, what kind of data we would need to produce. And we had actually, if we can go take a stroll down memory lane, that was Allergan in 2015, I believe, was getting ready to do the inversion with Pfizer to headquarter Pfizer in Ireland, and that we had conversations with Allergan into the lead up of that, and then those got shut down, obviously, through those conversations, and that ultimately was that inversion didn't go through. So, in the meantime, we had dosed that first patient. We were really excited by the data, and we circled back to Allergan. Uh, they were likewise excited by the data. They liked us preclinically. They liked us better once we had clinical data, and that's what led to the term sheet. So we'd had a couple of other groups that were interested, and had discussed some terms with, and ultimately the Allergan deal made the most sense for us.
What is the forum for those kinds of discussions? And I'm thinking of, a small biotech company who, as a good entrepreneur, as you've said, is thinking about, an exit for the company, whether it's, internal development and approval and commercialization or some kind of deal. I wonder, how is it happening like at conferences? You're making appointments with like the big players. Is there some other way that you're getting feedback from potential acquirers? Can you talk a little bit about just how those conversations happen?
Sure, yeah. I mean, it's a bit of everything, I think, Ben. So, I think every CEO, relies on their board to make introductions. Certainly, there's a lot of conferences that one attends. And there were several investment banking, biotech days that were also really productive. And so I think it's a function of getting in front of the, people on a consistent basis, familiarizing them with your science, your progress. Eventually, that leads to, the CDA, and if things are progressing as you hope, ultimately to the acquisition.
And the acquisition by Allergan was for how much?
So that was the headline value was 555 million.
Nice. Okay. And so, what circumstances led you to Immusoft after? I think that was your next company after Retro.
Correct. Yes. So yeah, just to put a little bit of the concept around what we were doing at RetroSense, it was delivering the gene using an AAV vector to the eye, intravitreal injection. What that means is, you're delivering a very small amount of vector, and so from a safety perspective, I had a lot of comfort with starting in an ocular setting. And so to put a little bit of, to add a little more color still, as I mentioned, this was all before the first gene therapy was approved in the Western world. So I was maybe taking baby steps, right? Let's get something to work in the eye before thinking about going beyond the eye and into systemic delivery with virus vectors. So once we were acquired, I went over to Allergan for a year to help transition that technology in. And I was really, thinking as my next act, I wanted to find a modality to deliver a gene therapeutic systemically that avoided a lot of the issues with virus vectors, namely the immunogenicity as well as immunosuppression requirements, and as well as some of the issues with gene modified stem cells, which are kind of the other primary modality for gene therapeutics. Um, and so, in the case of gene modified stem cells, there is, as you mentioned, a requirement for myeloablation, essentially chemotherapy that wipes out the bone marrow. That typically puts kids in the hospital for 4 to 6 weeks. There are a lot of morbidity issues associated, and unfortunately, all too frequently mortality associated with that. So, our approach at Immusoft, I probably jumped ahead a little bit, but through the course of that, scouring uh, the landscape for an approach that overcomes those shortcomings, I was introduced to Immusoft, and pretty immediately stricken by the elegance of the approach and how it overcomes some of those shortcomings.
So you agreed to join. How did you get up to speed after initially joining Immusoft as the new CEO? You joined as CEO, right?
I did, yes. So it was a trial by fire. My first day on the job was the day before JP Morgan, 2018. So I actually had done several months of diligence before I joined the company. and led some initial investment in the company as well. And Nonetheless, the founding CEO, Matt Schultz, I took his place, but one of the conditions was that he stayed with the company for a few months as to kind of help onboard me. So he very graciously abided, and that was really key to getting my arms around what is very complex but exciting modality here.
Yeah, well, let's talk a little bit about engineered B cells. Why is a redosing capability? And, we talked about freedom from immune suppression, myeloablation. Why is that so important for patients? I mean,
So
you mentioned it. I will say, with the myoablation ending up in the hospital and even death in some cases. So that's an obvious one. But maybe, what could you say about the avoiding the immune suppression and what that means?
So there's actually quite a bit of morbidity associated with the immune suppression as well. It tends to not get much attention, but it's certainly important. And uh, I guess again, unfortunately, there have been headlines of patients dying in some of these AAV clinical trials over the last year plus. And so being able to avoid those immunogenicity issues, I think, is really important. And so, again, as I was scouring the landscape, I was looking for that. And so the tie-in with redosing is because we don't have that, those concerns. It does give us the opportunity to go back and redose patients. And I think, if we contemplate a use case in particular, let's think, pediatric patients, as they grow, to be able to redose and have your therapeutic grow with them, I think is very important. And that's not something that is currently available with AAV approaches. And
Yeah, why can't you redose those?
Immunogenicity. So
Okay.
once you've done that initial dose, that patient has a pretty strong immune response to that vector. And so to go back and redose, I'll say with the asterisk, redose given current technologies, we haven't done that yet safely. And so to be able to ensure that you can redose, I think that's a big win for us and for the patients, for the community.
Right, right. And we're talking about engineered B cells. I mentioned in the intro, these are autologous. So you're taking cells from the patient, engineering them, and administering them back to the same patient, which is, I think, how you get around having to suppress the immune system. It's the patient's own cells. Can you just describe maybe at a high level, Sean, what that process looks like and maybe why it hasn't been done before?
Sure, yeah. So the process, we start with an apheresis, which is it's not a blood draw, but you take the patient's the immune cells effectively from the patient's blood, and you replace the plasma, um, and so that's our starting material. And from that, we isolate out those B cells that are ultimately this the next point in the starting materials. So in culture, we introduce our gene of interest. We use a transposon system, sleeping beauty transposon, and that's very effective with B cells. We've found it to be better than most virus approaches, and importantly, I'll add that it's a fraction of the cost. So
Hmm.
I'm getting off topic a little bit to scalability, but I think that's a big important part of the puzzle that we've prospectively addressed as well. But So getting back to the approach, we then differentiate the B cells, we expand them, And so we differentiate toward that plasma cell state. And the plasma cell, we sometimes use that interchangeably with B cells, but it's an end-stage differentiated B cell, and it's really this powerhouse biofactory of the immune system. So any given plasma cell is producing and releasing thousands of antibodies per second into the circulation, and we harness that ability of the plasma cells, but we direct them when we modify them genetically. We direct them to produce therapeutic proteins of choice. And so that gives us really, we think a broad opportunity to address a multitude of different therapeutic indications. So we're just getting started, seeing great results so far in the clinic, and so this is very exciting.
You mentioned scalability as a kind of attribute for this process, and I'm curious. Well, I have two questions. One is, what is the time period from when the cells are extracted from the patient and then delivered back in? And then second, is that done on site, at a clinic, or are the cells mailed and then sent back? How does that work?
So, yeah, the patient experience, they come in for that apheresis. They go home, we take that apheresis product. Our culture system is 1 week. And so another factor in scalability, the shorter the better. There's a lot of person hours that go into the culture, the time in culture. So 1 week is a nice short time frame. But from there, we do sterility studies on the drug product, and we actually do have about 3 weeks of sterility testing. And then we ship that back to the patient or pardon me, back to the hospital. And that's where the patient had that apheresis done. So the patient then comes back in and gets the infusion. The infusion takes an hour, which, is for our lead indication, that's actually uh, surprisingly short for these patients because they're used to coming in, 4 hours every week. And so 1 hour, for a protein biofactory that might be delivering that therapeutic for, years, that's a big eye opener for these patients.
Yeah, and you're referencing enzyme replacement therapy for MPS1, which is your lead indication. Your lead candidate is ISP001, a rare and progressive, I think most of the time, inherited genetic disorder. What can you say, Sean, about the choice to pursue MPS1 as your lead development program?
So we actually, put a ton of thought into that. So appreciate the question. We started with north of, 100 indications where we thought we might have application with the B cell approach. And so, the initial cutoff is it a protein therapeutic that can be delivered in trans? And that's what kind of got us to, 100 indications. From there, we looked at 11 different aspects from clinical trial complexity, patient population, uh, proof of concept, whether there's already human proof of concept for the existing that existing protein that we're ultimately looking to deliver, intellectual property. So we rank ordered across 11 different um, features, and ultimately MPS1 rose to the top. I left out unmet need. There's actually still a pretty significant unmet need, and despite a current enzyme replacement therapy that's on the market.
Right, and you talked about, the patients having to do multiple hours of administration for an ERT. I, MPS1 is a lysosomal storage disorder, which was, an kind of an early focus area for biologic companies like BioMarin. I know was a kind of early leader in that space, creating these ERTs. Aside from just the kind of sheer administration convenience associated with engineered B cells. Are there other ways that this approach could improve upon ERTs?
We do believe, yes, indeed, that we can ultimately, um, let me rephrase that we may ultimately provide benefit above and beyond what ERT can deliver. And so in the case of the current standard of care, it has a plasma half-life on the order of a couple of hours. And so that's part of the reason for the need for weekly 4-hour infusions is to try to keep that enzyme level up. In our case, we're delivering 24/7, and we ameliorate or eliminate that sawtooth effect. By sawtooth effect, that that spike and trough that is observed with pulsatile infusions. So the expectation is that 24/7 delivery of that therapeutic protein may ultimately yield better outcomes in these patients. So,
Got it. Got it. And so what would you anticipate being the kind of dosing schedule for this product in MPS one, I guess compared to the weekly 4-hour administration that's more or less the standard of care now, I think.
yes. So we're actually exploring an annual dosing regimen.
Annual,
Um,
okay.
yeah, yeah. So we think that would be obviously a huge improvement over weekly infusions. Uh, we did redose our first patient, and that was after 18 months' time. So he received the lowest dose that we expect to give in this trial. Um, Saw some I'll go ahead and say very compelling signs of efficacy, including a 240-meter improvement in his 6-minute walk test. That was at a year. Um, he saw pharmacodynamic improvements as well as patient-reported outcomes. Importantly, very significant reductions in pain. So it's just one patient, but I think that having three, from the pharmacodynamics, the functional outcomes, the patient-reported outcomes, all pointing in the same direction is super exciting. We have left that patient on standard of care. So that was part of our agreement with the FDA is that we'd start out by dosing with ISP 001, but keeping the patient on standard of care. We removed that, we referred to it as an enzyme holiday. So he had an enzyme holiday for about a month. We saw those functional improvements continue forward, which was really exciting. Some of the biomarkers showed changes, which one would expect when removing standard of care. But we thought that it would be important to go back and redose that patient, and with the objective of ultimately taking him off of standard of care.
Right, right. Just looking through your pipeline, you've got several assets that are in the discovery phase, I think, right now that target larger therapeutic areas in patient populations, areas like Parkinson's disease, obesity and diabetes, oncology, autoimmune diseases. Can you talk a little bit, Sean, about Immusoft's clinical strategy and kind of how you're thinking about progressing other assets into clinical studies, as a kind of second wave to your lead development program in MPS one?
So yes, yeah, we actually took a two-part approach initially toward a platform consideration. And so what I mean by that is our next program in line is MPS 2. And so the strategy there is with some safety and preliminary signs of efficacy, we went back to the agency and we requested a waiver for definitive tox studies for the MPS 2 program. And so they were amenable to that. And so we think that's a big win. We don't have to kind of reinvent the wheel every time and do, these sprawling and expensive toxicology studies, which means ultimately we think we can accelerate, the next program and the next one after that to the clinic, which will yield the best outcomes more broadly. So I mentioned our kind of pipeline prioritization strategy. We actually had a lot of lysosomal storage diseases that popped up on our radar, and in no small part because there are existing programs on the market
Right.
that are delivered, that have that same um, call it issue with durability in terms of the half-life, and so the requirement for frequent redosing. Moreover, they're genetically defined. We know exactly what protein to deliver and why. And so the plan has been to start there, build that clinical experience, and then move from there into um, I think of them as frankly higher risk but higher reward opportunities in CNS, autoimmune, oncology, etc.
Yeah, and I mean, I've read that there's somewhere between 50 or maybe 70 lysosomal storage disorders. What makes you want to not just pursue those? You got plenty to choose from in that kind of defined category, but you're more ambitious. You're looking at some of these really broad and large disease areas with huge unmet need. What would you say about that?
So yes, we actually it sounds like you were in the boardroom with us because we had a lot of conversations on that very topic, and we still do. But what we ultimately decided is, it makes a lot of there's so much application across this platform that we would be doing ourselves and probably so many others a disservice by not looking to advance in some of these other more ambitious therapeutic areas. So what we're actually working towards is establishing some early POC in some of those higher risk, higher reward indications and looking to partner on those. Uh,
Okay.
yes, yeah. While continuing forward with the lysosomal storage approaches, which generally those are orphan indications.
Right.
Um, many of them a billion plus opportunities. Um, and but continuing forward with those, under the auspices of Immusoft and kind of bringing, ultimately with the ambition of kind of bringing it all forward and having partners for many of those programs, but working towards the clinic with several other lysosomal disease approaches.
And with your MPS one program, which is your lead, and then your follow-on project or program in MPS two, are those being developed completely in parallel? Do you wait until MPS one reaches a certain clinical milestone before then progressing MPS two? How do you think about that, managing that?
So we are, we've had a lot of dialogue with the agency on MPS 2. We think that, we've got just a couple of other blocking and tackling items to be able to move that program forward into the clinic. Right now we are focused on MPS one. It's in the clinic. We want to continue forward with that. And as we go out and secure more resources, we'll look to advance MPS two into the clinic.
Got it. I want to follow up on some of your comments about your engagement with the FDA and kind of get a sense of that. But before I do, Sean, you're CEO and chairman of the board at Immusoft. You're also a board member at Ray Therapeutics and at Opus Genetics. What could you say about the most important role that a board member can play at a company?
Wow, that's a very broad question. I think do no harm is one of the one of the most important things, which seems obvious, but isn't always so much in practice. But I think the as a board member, you've got a fiduciary responsibility to the shareholders, which means you've got to work closely with management to understand the direction of the company, the strategy, and when and where the decisions to deploy resources come into play, and ensure that that's all um, well designed and strategically thought through. And then above and beyond that, bringing resources to the table, be they, potential investors, strategic partners, new employees, advisors, etc. I think those are all important parts of being a good board member.
Are there any downsides to being both the CEO and the chairman of the board at the same company? And I ask because, I've had guests talk about, difficult conversations with, the CEO between the CEO and the board where there's disagreement. You're CEO and chairman, so you're probably not disagreeing with yourself a whole lot. But what would you say about you know, being both, serving on both, and are there new challenges associated with that?
So at the end of the day, whether you're board chair or not, your vote counts, the same as any other board member. So, as CEO, I'm wearing the hat of the operator, by and large, coming into the board meetings, and it's always important to me to coordinate with the board before ever getting into the meetings, and so there's very few instances where something really comes up at the board meeting that's a surprise to anybody.
Right. Okay. Let's get back to FDA. The current administration and FDA seems fairly engaged with gene and cell therapy, particularly in rare diseases. We've seen, just as an example, the recent idea of a plausible mechanism pathway floated for personalized therapies that would potentially remove the requirement for randomized controlled trials in some instances. ISP 001, your lead candidate, received fast track designation from the FDA, I think in October. What more could you say, I guess, about your recent interactions with the FDA? What other color could you add there?
Thanks. So I think that the agency is actually doing some very innovative, taking some very innovative steps towards removing some of the barriers for cell and gene therapies. The new approach that you mentioned, that's in I think really an offshoot from the baby KJ
Right, exactly. Yeah.
dosing, which was really extraordinary, and just such an amazing collaborative effort and story behind that. So for the FDA to take a close look at that and start to build in mechanisms to enable more of those approaches to move forward, I think makes a whole lot of sense. And if you dig in, I mentioned platform consideration, platform designation. I think a lot of that actually plays into the baby KJ story as well. That if you've got a platform that's that's working from a delivery perspective, and you're targeting a known gene, um, to be able to make a one-off or small n off and take those into a clinical setting early. I think that's just a huge win for for everybody. And beyond that, the agency in September came out with guidance for orphan diseases as well that provide a pathway for single-arm clinical studies. So I think that a lot of that is um, innovative, helpful, and for orphan cell and gene therapies in particular, those are nice alignments from a regulatory perspective.
Have you noticed any changes in style or strategy or even performance, good or bad? Maybe thinking back to your time engaging with the agency at RetroSense, kind of up through Immusoft. How has the agency evolved in your view?
So I'll avoid any political commentary because that's there's obviously been some changes that are associated with the political changes, but I guess what I'd say is going way back to the early RetroSense days, evolved is the right word. That the agency has evolved with the science, and I think that's been really important. We're doing things now with so many different gene editing, gene therapy, cell therapy modalities, CAR T, you name it. I think a lot of the old frameworks just would never be able to keep up, and so um, there has been a fairly consistent evolution, and these last two changes that I mentioned, they seem to be both steps in the right direction towards keeping up with that evolution.
Yeah, and I think you engage with Sieber for your products. Richard Pazder now is going to be leading Sieber, I believe. Is that correct? You'll be working with his group.
My understanding is he's leading Cedar.
You're right. You're right.
Yeah,
Cedar. Yeah,
yeah. So
that's correct. Yeah. Okay.
Dr. Prasad is Sieber.
Yeah. Thank you for that correction. Is there anything else, Sean, that you would say, kind of looking forward in terms of, and maybe even not specific to the FDA? I don't know how much you're thinking about global markets, although you probably are, given your background and work and kind of cross-border corporate development. But is there anything else you would say about the regulatory outlook for cell and gene therapy next year? Is there anything you'd like to see that you're not seeing yet?
So yeah, I appreciate the question. Um, there's still a boy, what am I thinking of? The rare pediatric disease voucher.
Yeah.
That's a program that I'm hopeful gets re-implemented soon. I think there's a pretty strong sentiment that will be re-implemented, but it can't come soon enough. It's something that is just so important to those of us working in pediatric orphan diseases. And so, to give a little color for those who may not be familiar, that voucher program, upon approval of your rare pediatric indication, there's a voucher that allows for accelerated approval of another program, and those can be sold on the open market. The last few that I'm aware of have been purchased for on the order, of $150 million. And so for these, again, rare pediatric diseases, that's been a huge part of the decision-making process, and it's a big part of the value proposition. So I actually sat in on the ENC committee hearing a couple of months ago. I got voted 24-0 to, to advance bipartisan support. I think there's general consensus that that's going to come through and, hopefully soon.
Yeah, that's a really important incentive. I'm glad you mentioned it. I think that program expired, I want to say like last September. Has it been that long that it kind of sunsetted and has not yet been reinstated? I know there are a lot of folks out there that want that incentive to come back. Yeah, and you mentioned you can use the priority review voucher. The going rate is somewhere north of $100 million recently, and it's kind of been that way for a little while. But that voucher can be used for any development program. That priority review, it doesn't have to be for the purchaser of the voucher program. They can use it for any development program, which becomes quite important, if you're developing a product and you have some close competitors and, are kind of trying to be first to market. It's a valuable incentive and a valuable tool. So I think, I'm with you in hoping that one comes back. We're getting to the end of our time, Sean. I wanted to just end with, kind of your top priorities, what's next for Immusoft, what you're most focused on right now.
So we're still, driving forward with our lead program clinically. So MPS one. We did have a recent meeting with the FDA, and that was important in gaining alignment on what that pivotal trial looks like. Without getting into the weeds, we do have alignment on the size of the study, the pediatric population, the endpoints, and so those are super important things for advancing the program from here. And in the meantime, we keep our nose to the grindstone. We got a really great group of scientists that are continuing to produce amazing preclinical data, and so we're always working to advance those follow-on programs as well.
Yeah, and given that you're working in pediatrics, would you be, assuming that priority review program, voucher program is reinstated, would you potentially be eligible for that with any of your clinical programs?
So MPS one, absolutely. Yeah,
Yeah.
thank you for asking. So we've actually got a rare pediatric disease designation already.
Okay.
And so that is what enables you, upon approval, to get that voucher. MPS two, we believe, would also be available and or eligible. Pardon me. So that's across a number of lysosomal diseases. They're almost because they're inborn
Right.
errors. They're almost all pediatric.
Yeah, that'd be pretty nice, I think, to get somewhere over $100 million, no strings attached to bring back into the company and
Right.
maybe tackle some of those even more risky assets that you have in discovery right now.
Right, right. Definitely fuel more innovation.
Absolutely. Well, Sean, thank you so much for coming on the show. It's a real pleasure speaking with you.
Likewise, my pleasure. Thanks for having me on the show, Ben.
We've been speaking with Sean Ainsworth, a CEO and chairman at Immusoft. I'm Ben Comer, and you've just listened to the Business of Biotech. Find us and subscribe anywhere you listen to podcasts, and be sure to check out our new weekly videocasts of these conversations every Monday under the Business of Biotech tab at Life Science Leader. We'll see you next week, and thanks as always for listening.